for the opportunity to talk my present research in this honorable conference. My name is Kazuo Ishii from Tokyo University of Agriculture and Technology. Today, I would like to talk about data analytics and mathematical modeling for psychiatric diagnosis in a big data processing environment. This is the agenda for today's presentation. Next slide, please. Emergence of the next generation sequencing technologies and its application to clinical and biological research leads to the era of genomic big data. The cost of human genome sequencing is lowering year after year, and its speed surpasses the Moore's law after 2005. Today, realizing the personalized medicine with big data is a fascinating topic in the current clinical research. In the clinical studies, big data analysis is very popular in cancer and cardiovascular diseases, but not in the neuropsychiatric disorder because of its ambitious causality and the difficulty of obtaining the specimen samples. In Japan, neuropsychiatric diseases such as depression and bipolar disorders are increasing year after year because of bad economical status of Japan in decades and the natural disaster, including the big earthquakes. Diagnoses of neuropsychiatric diseases were performed based on only the interview with the doctor, by the doctor at the clinical site. And there is no evidence-based disorders, diagnosis for neuropsychiatric diseases, such as biochemical and molecular biological tests. Therefore, big data-based new diagnosis system using genomic big data is expected to provide revolutionary innovation in the mental health. Okay, the aim of our research is the development of big data mining method that is in development of optimized algorithm and mathematical modeling method with genomic big data. We use 500,000 to 10 million exploratory variables obtained from NGS and microarrays. We observe the suitable diagnosis markers and create the optimized and mathematical model for neuropsychiatric diagnosis. Our research target is focusing on the three major mental disorders such as depression. In this slide, the outline of the analytics process was shown. Six processes were contained in the overall step. Unstructured data, unstructured data, structured data, selection, discrimination, mathematical modeling, optimization, and evaluation. Processing unstructured data containing map, Hadoop map reduce, shell scripting, and data processing with no SQL, Monte Carlo simulation. Processing structured data contains data processing with RDMS, MySQL, and PostgreSQL. Selection of explanatory variable statistical containing statistical hypotheses, testing, such as student t-test and monthly t-test. In the discrimination process, containing variety of data mining methods, such as match variable analysis and machine learning. In the mathematical modeling process, we use linear regression model and logistics regression model and mixed model. In the final step, mathematical models were evaluated by cross-validation, including leave-one method, leave-one-out method. This is a comparison between HPC, high-performance computing, and AWS cloud. AWS produces a larger memory. HPC produces a larger memory than AWS. Both platforms have different character. Therefore, platform should be selected based on its purpose. Here I show the threshold methylation coding software, Bismarck. Bismarck is based on the mapping of bowtie. Because Bismarck and ours is a larger memory, very large memories, such as Bismarck requires 870 gigabytes in one process. Ours requires 900 gigabytes in one process. Amazon Cloud could not analyze methylation coding with Bismarck. This is our test with HPC and cloud computing. Here I only show the practical test of data mining using microarrays. Result of NGS will be shown elsewhere because of preparing patent pending. In this result, Illumina 450K methylation microarray was used for the structured data. Mann-Whitney U-test and its ranking were used for the selection of explanatory variables. Linear discriminant analysis LDA was used for discrimination of data. Discriminant function was used for mathematical model. Backward elimination method was used for optimization of models. Cross-validation and returning set and validation set was used for evaluation of model. DNA methylation rates was evaluated by beta value. Beta value is calculated based on the ratio each methylated site and each sum of the methylated and unmethylated sites. This is the graphical data of the beta score. In the horizontal axis, the beta score is indicated. In the vertical axis, frequency of the methylation site indicated. By this visualization, you can find that DNA methylation rates did not show a normal distribution. Okay. This is the example of one neuropsychiatric disease. 20 patients and 19 healthy volunteers were tested with 500,000 explanatory variables with Mann-Whitney U-test. The significance of the markers was selected by p-value, and 362 methylation sites were selected for explanatory variables. Linear discrimination analysis is multi-variable analysis to discriminate samples to two or more categories with multiple variables. The criterion of discriminant analysis is modeled by discriminant function. In the next slide. Discriminant function is a linear discriminant function consists of explanatory variables. Discriminant function gives discriminant core to discriminate the sample. Next slide. The discriminant efficiency was evaluated by sensitivity and specificity. Sensitivity means the ratio of positive patients in all patients. Specificity means ratio of the negative healthy volunteer in all healthy volunteers. Okay. This is the evaluation result using a training group by discrimination analysis with top 20 significant methylation sites by Manhattan test. Discriminant core were plotted into the scattered plot. In the training group, discriminant analysis could completely discriminate between 19 healthy volunteers and 20 patients. Upper left area of the estimate. Upper left area is the area of estimated diagnosed negative, and the lower right area is the area of the diagnosed positive. You can achieve the perfect discrimination model in psychiatric disorders. This is the evaluation result using the validation group by discrimination analysis with sample with the same top 20 methylation sites used in the training group. Discrimination score were plotted into the scattered plot. In the validation group, discrimination analysis could completely discriminate 12 healthy volunteers and 12 patients. Upper under left area is the area of estimated diagnosed negative, and lower upper right area is the area of diagnosed positive. This discrimination model shows the perfect discrimination efficiency in the validation group. This is the last slide. This slide shows the hierarchical clustering using the 12 significant methylation sites by training group. Hierarchical cluster, healthy volunteers and patients were perfectly separated with the hierarchical clustering with top 12 significant methylation sites. Left cluster is patient, and right cluster is healthy volunteers. In summary, we are analyzing big data clinical data from microarray, next-generation sequencer, and metadata. For methylation analysis required extremely large memory in the analysis. Amazon Cloud could not analyze the big-scale methylation data, which required an almost 1 terabyte memory. In this case, we should need special strategies by the distributed file system and the distributed processing system. So, big data processing environment should be selected based on its performance and purpose of big data analysis. For the development of the diagnosis system, selection of non-parametric testing and the discrimination of discriminant analysis is very effective. It achieves the perfect discrimination between patient and healthy volunteers. Thank you for your attention. Thank you.
It's an impressive study, and I wonder why methylation data was selected to do this study, because the data amount is so large. On the other hand, you have other more common platforms, for example, at the gene expression level, at the protein level, or at the mutation level. Why? I just wonder why methylation data was selected to do this.
Okay. Sorry. Tell me again. I am disabled and with communication like this, and then tell me again. Sorry.
Louder enough? Yeah. Why did you use methylation data to do this study instead of using more common platforms, for example, genes? At the gene level, you have only about 20,000 predicting variables, and in this case, you have as many as 10 million. So why do you choose methylation?
Methylation. About methylation, sorry. What kind?
Actually, I think I can answer that question.
Okay. We tried a lot of analysis using expression, and methylation. Methylation analysis has more valuable numbers than expression analysis, and neuropsychiatric disease. We don't know why we affected neuropsychiatric disease. We don't know the we don't know the biochemical and molecular biological basis about neuropsychiatric diseases. But there are some evidence methylation ratio level affected by neuropsychiatric diseases, and then we select the methylation. We also try to analyze the expression level, and then finally I use the analysis with methylation, expression, and other information. But now our first try is DNA methylation. This is the first trial.
Yeah, I'd like to know, you use the Apache Hadoop to parse the whole data set that you have. Did you use the recent release of Hadoop, or you modify it to your purpose when you train the algorithm on it? Apache Hadoop, that? Okay, you use the Hadoop algorithm for data analysis. Yeah, so you use as it is from Apache server, or you did modify it for your purposes? Because the open source algorithm.
We tried Hadoop for the simple calculation, like the pig Latin, and then I used I made the calculation like the self-scripting, and it is difficult to perform the complex analysis using Hadoop, and then I use Hadoop using simple manipulation of the data from unstructured data. Unstructured data, we first specific pattern and specific simple counting. After the simple counting, I can perform the statistics analysis and character-based analysis.
Thank you.
Yeah, we'll move forward. Thank you very much.